Hi there, good afternoon everybody. Welcome once again to one of our CRIC webinars, and today we're going to be talking about imaging flow cytometry. For those of you who don't know me, my name is Derek Davis. I'm the Science Technology Platform Training Lead here at the Francis Crick Institute in London. For many years I ran the core flow cytometry facility here, and you can follow the facility on Twitter at CRICFlow, or you can follow me on Twitter at CRIC Training. Actually, I say we. I'm not going to give this webinar today. I'm going to hand over in a moment to Kevin Rees, who's a senior laboratory research scientist in the Crick flow cytometry facility, and he has a lot of experience in imaging flow cytometry, having done a lot of it during his PhD at QMUL before he joined the lab here at the Crick. You probably saw from the splash screen when you came in that there are a couple of buttons you can use in Zoom. There's a chat button, so if there's an issue with sound or vision, please put that in there and I'll try and sort that out as we go along. If there are any questions, there's a Q&A button as well. So if you type in your questions there, if it's relevant, we'll try and answer those as we go along, but otherwise we'll try and ensure that there's enough time at the end to cover the questions that you have. This webinar is also being recorded, so at the end I'll be able to send you a link to the recording as well, and right at the end I'll also give you another link to many of the other videos that we've made. Videos, that shows my age a little bit. Webinars, I think. So I'm going to hand over to Kevin now, and he's going to talk about imaging flow cytometry.
Great, thank you very much, Derek. So thank you everyone for joining us. So as Derek said, my name is Kevin and I work in the flow cytometry core facility at the Francis Crick Institute. And I'm going to give you an introduction today to imaging flow cytometry. So I'm just going to give a brief overview, an introduction to what I mean by imaging cytometry. And then I'm going to talk a little bit more in detail about a particular imaging cytometry platform that we have a lot of experience with at the Crick called the ImageStream. And then I'm going to talk a little bit about some best practices for sample preparation and acquisition for an imaging cytometry experiment. Talk a little bit about data analysis and show you some example applications and some data that's been generated by people at the Crick. Okay, so firstly, what do I mean by imaging cytometry? So imaging cytometry, or specifically imaging flow cytometry, occupies this space between fluorescence microscopy, where we generate images of cells, and flow cytometry, where we use a fluidic system to focus a large number of cells per second through a series of illumination points to get single cell data. So imaging cytometry combines these into a platform where, again, we're focusing single cells through a series of illumination points, interrogation points. But rather than just digitizing the intensity of the fluorescence or the scatter into a single value per cell, we're actually imaging those cells. So not only do we get information about the intensity of our fluorophores, we also get the spatial information as to where in the cell those fluorophores live. And so if we take a little comparison between microscopy, flow cytometry, and imaging cytometry, if you're doing a microscopy experiment, the typical number of fluorescence parameters that you'll be limited to is 3 or maybe 4 or 5 if you've got a particularly good microscope. Whereas in conventional flow cytometry, we can push this to 18 colors plus quite easily nowadays. And imaging cytometry, particularly the ImageStream platform that I'll talk more about today, you have the choice of using up to 10 fluorescent channels, although in reality, going above 7 or 8 can become quite challenging. In terms of throughput, so microscopy, depending on the size of the cells and the magnification that you're using to image, you're probably limited to maybe 100 or fewer than 100 cells per field, meaning that actually it's quite difficult with microscopy to get statistically robust numbers of cells recorded for your sample. Whereas, of course, flow cytometry doesn't really have this issue. It's quite easy for us to record thousands of cells per second to generate data files containing millions of cells. And imaging cytometry, perhaps we can't usually go as fast as most modern conventional cytometers, but we can certainly image hundreds of cells per second, which again means that we can create these robust data sets that have large numbers of cells in them. When we talk about getting spatial information from our cells, of course, microscopy wins here. So if you need to image extremely small structures or resolve very small structures, then most microscopes can go down to nanometer resolution, depending on how much you paid for your microscope. And of course, we don't really get any spatial resolution with conventional flow cytometry. The only exception being really is if you do some kind of FRET assay, but then even then you only know that your probes are in close proximity with each other, but you don't know where in the cell they are. Whereas with imaging cytometry, of course, we do get the spatial information. We can see where the probes are within the cell. But the best that we can do with the image stream platform is down to 1/3 of a micron square per pixel. Of course, with microscopy, we're not analyzing cells in suspension. They're cells that are either adherent or put on a slide and cover slip. Whereas flow cytometry and also imaging cytometry, we are imaging cells in suspension that are flowing through a flow cell. So why might somebody want to use flow cytometry for their particular experiments? So the advantages over microscopy. So firstly, we can collect data in an objective way. So it's quite easy for researchers, even unconsciously, when they're using microscopy, to select cells to image that either support the question that they're trying to ask, or that look good. It can become a little bit subjective. Whereas with the image stream or with imaging flow cytometry, we're able to image every single cell that flows through the illumination points. So we're able to collect the data in an objective way and not just select for the cells that look good. It is because we're using hydrodynamic focusing, just like in conventional cytometry, to focus the cells single file through the flow cell, through the interrogation points. We can collect images in a very high speed way and collect large data sets. And so we can create files that give us statistically robust numbers of cells to analyze. We can image multiple fluorophores simultaneously. So with most fluorescence microscopes, you sort of select a filter block, take an image of your GFP, for example, and then slide the filter block for the DAPI in place and take that image. Whereas in an imaging flow cytometer, all of your emission wavelengths are collected and imaged at the same time. And we can push the number of fluorophores or fluorescent probes that we can use in our experiments. So we're not limited to maybe 3. With the image stream, you can use potentially up to 10 in one assay. Compared to conventional cytometry, of course, the advantage is really that we get high content data. So we get spatial information. So we can get information about the shape of the cells, and also the location of probes within the image. And the images are always linked to the quantitative or the numeric features that are extracted from those images. So, for example, all of the features such as the size or the intensity of a particular probe can be plotted in a 2D scatter plot or as a histogram, for example. And you can click on an individual data point on that scatter plot, and the software will show you exactly the images corresponding to that particular cell, and the vice versa. So if you see the image of a cell that looks interesting, if you click on that image, then it will immediately show you on your scatter plots where that cell exists in the data, which is a nice way of being able to link and identify the numeric data to the images. But maybe why would imaging cytometry not be appropriate for your assay? So disadvantages over microscopy. So the pixel resolution isn't as good as you can achieve with a good high resolution microscope. So the best that we can do is 1/3 of a micron square per pixel, which means that if you're trying to resolve any structures in a cell that are closer together than this or as far apart as this, then you're not easily going to be able to, or you're not going to be able to resolve them using imaging flow cytometry. Of course, you can't re-image the same cell like you can in microscopy if you're doing some kind of time lapse or looking at how some process changes over time. You can't re-image the same cell. Once it's passed through the interrogation point, it's gone to the waste. And the imaging is not confocal. So you're not taking a super fine focal plane through the cell like you can using a confocal microscope. The best that we can do on the highest magnification that we have is a 2.5 micron wide focal plane through the cell. And of course, as we're flowing cells through a flow cell, it's only applicable to cells in suspension. So we're not able to get any information about tissue architecture or how cells interact with each other in a tissue. You would need microscopy for that. And disadvantages over conventional cytometry. One of the main ones is slow flow rate. The actual acquisition of samples itself is slower, the flow rate, and also the time between samples is quite slow. So the image stream platform that I'll talk about later on is the sample is driven through the flow cell by a syringe. It's a volumetric system. And the advantage of that is that it will actually tell you the concentrations in terms of cells per mil of each of your populations. But the downside is that it's quite slow to load and unload samples. Your fluorescence resolution is probably mostly or usually slightly poorer on the image stream compared to most modern conventional cytometers. So if you have a positive and a negative population that you're unable to completely resolve on a conventional cytometer, it's very unlikely that you're going to be able to resolve them on the image stream. So your resolution is slightly poorer. And because we're not just generating numeric, single numbers to summarize our data like we do in conventional cytometry, we're actually generating images. And for every cell that you record, you also record the number of images depending on the number of channels that you have open. The file sizes become very big very quickly. And so an imaging cytometry file with, say, 200,000 events could take 5 minutes to open, for example. That would be quite a large file. So that is another downside. Depending on the goal, your analysis can end up being quite complicated, and often the acquisition of samples takes less time than the actual analysis, depending on what you're doing. If you're doing something very simple, it may not take particularly long, but I find that most analyses that we do, it's a long process of making sure that the analysis that you've done is appropriate to answer the question that you've set out to answer. So when we have new users come to the lab and they hear about our imaging flow cytometer and they're interested in using it because it sounds quite cool, we usually go through a sort of set of questions that we ask them to see actually whether this is appropriate for what they want to do. So the first question I would ask is, do I need to see either the tissue architecture or the cell ultrastructure, so the very small organelles in the cell? And if the answer to that is yes, then microscopy is the set of techniques that you're going to need to use. But if no, the next question would be, well, do you need spatial data? Do you need information about either cell shape or location of probes? And if the answer to that is no, then you should use conventional cytometry because your acquisition will be much faster, your files will be slower, and your fluorescence resolution will probably be better. But if you do need spatial information, the next question is, do you need a statistically powerful number of cells, or are you just looking to get an image to sort of perhaps understand or get an image of a process that's going on in your sample? And if the answer is no, then fine, you can use microscopy to take an image. But if you need thousands or hundreds of thousands of cells, then imaging cytometry is the platform that you need to use. So there are a number of imaging flow cytometer models, and some of these, like the CytoBoy or the FlowCam, are mostly marketed towards marine biology labs. And these systems, so DEPARray and CellM1, are sorters. So they sort cells, single cell, into different plates, depending on their morphological features, so their size, for example. But I think most flow cytometry labs, when they talk about imaging flow cytometry, are either talking about the image stream or the flow site from Luminex. And it's mainly the image stream that I'm going to talk about today, because I think that's the main instrument that people use when they need a high performance imaging cytometer. The flow cyte is the baby cousin of the image stream, and the only main difference is that the flow cyte is limited to 20 times magnification, whereas the image stream can do 20, 40, or 60. Okay, so a brief overview of the image stream. So it comes in two options, either a one camera model or a two camera model. And I'll explain a little bit more about that as I go on. But if you have the two camera model, which most people probably end up getting, you have up to 10 fluorescence channels. So the image stream can image up to 10 fluorescent channels and two bright field or transmitted light channels as well. And you also have the choice to image the scatter or the side scatter of the cell as well. You have the choice of using either 20, 40, or 60 times magnification. And this is not something that is fixed by an engineer, for example. So you can easily change between 20, 40, and 60 quite seamlessly, depending on your needs. And the pixel resolutions here correspond to the different magnifications. So at 20 times, 1 pixel represents 1 square micron. At 40 times, you have half micron resolution. And at 60 times, you have third of a micron resolution. And there are various laser options that you can have from Luminex. I think the most common ones are probably the 488, 561, 642, and the 405. That's what we have in our instrument. So it's a 4 excitation laser instrument. But you can also have a near UV 375 and a 592 as well. And there is also an option to upgrade your blue laser to have a high power laser as well. But this is only really useful if you're either doing something like small particle work or if you're looking for very dim signals. So this is what the optical bench looks like for the image stream. So you have a flow cell here where the cells are hydrodynamically focused through 2 interrogation points. So the image stream has 2 optical paths. The 642 and the 405 lasers are aligned to 1 optical path. So these are collinear. And the 488 and the 561 are aligned to the other optical path. And these 2 are collinear. And so the cells are illuminated by the lasers, and they're also illuminated by an LED system that creates the bright field transmitted light system. And the image stream has no forward scatter. And we don't really need one because we have bright field images, so we can get things like shape and size information from the image. The light is collected by one of the 3 objective lenses, so 20, 60, or 40, and these literally just slide back and forth on a rail that you control through the software. And then the 2 optical paths are split, where one optical path goes towards one camera, and the other optical path goes towards a different camera. So the image stream doesn't use photomultiplier tubes or PMTs as detectors. It uses these 2 CCD charge coupled device cameras. And the 2 optical paths are split up into their various bandpaths of light by a set of filters that are held at angles. So that different wavelengths are sent onto different parts of the camera chip. And so this table sort of shows the band passes of the various channels. So on camera 1, we have channels 1 to 6. So we have these 6 regions on each camera. And you can see that each region receives light from a particular bandpass of emission. And we have channel 1 reserved for a bright field transmitted light image, and also channel 9. Although if you have particular needs, then you can change those bright field channels. And also we have the option of including a side scatter or dark field channel as well. Okay, so these cameras work in a mode called time delay integration. So the surface of a CCD camera is made up of these small photoreactive cells. And you can kind of imagine each cell sort of representing a pixel. And when photons hit one of these cells, then the cell becomes charged proportional to the number of photons that have hit that cell. But we're trying to image objects that are moving quite quickly. And so the system uses a process called time delay integration to prevent us from getting blurry images from these fast-moving objects. So if you imagine a cell traveling through the flow cell being illuminated, and say the cell has GFP and DAPI staining, for example. So the set of filters will reflect the different band passes of light onto different portions of the cameras. And so you can imagine this GFP end up being reflected onto this strip of this camera. And while the cell is in this position, the number of photons hitting this cell here will accumulate a particular charge there corresponding to the brightness coming from that portion of the cell. And then the charge in this cell is transferred to the next cell below it in exact synchrony with the rate at which this cell is traveling through the flow cell. And then this cell continues to accumulate charge. And then as the cell moves on, that charge is transferred down and down and down. And the effect of this is that light coming from a particular part of the cell is accumulating for the length of time that the cell is passing through the flow cell. And that eventually when the cell passes off at the end of the camera, you get this deconvoluted image where the brightness assigned to this particular pixel here has accumulated over the length of time that the cell was traveling through the flow cell. And this achieves two things. So one, it means that we're collecting light over a much longer period of time. And so because of this, the image stream is very good at picking out very dim objects from background. And the other benefit is that we don't get blurry objects. So if you imagine that you have a cyclist whizzing by, if you hold your camera static and wait for the cyclist to come in front of your lens and then you take a photo, you'll have a very blurry cyclist. Whereas if you move your camera at the same speed as the cyclist and then take a photo, you'll get a nice sharp image. So time delay integration is kind of a digital version of moving your camera lens with the same speed of the cell. And so this is the kind of image that we get out of the image stream. So these are images corresponding to one cell. So this is a bright field transmitted light image of the cell. This is maybe a GFP or a FITC or something. And this is probably something like APC. And this here in channel 6 is what an image of side scatter looks like. And here in the software, we just have an overlay between the channel 2, the FITC, and the channel 11, the APC. And every pixel is digitized into a 12-bit number. So what that means is that every pixel has a maximum value of 4096. So every pixel has an intensity between 0 and 4096. And if you take one of these images, then you can think of it as the whole image being broken up into a series of pixels where each pixel has 3 values associated with it. It has its X coordinate, it has its Y coordinate, and it has its intensity value, its brightness, which you could think of as a Z coordinate. And so when we're analyzing our data, we use this combination of the X and Y, which gives us the spatial information, and the Z value, which gives us the intensity value. Okay, so let me talk a little bit about sample preparation and acquisition. So when you're thinking about building a panel for an imaging flow cytometry experiment, a lot of the considerations you need to make are similar as for conventional flow cytometry. So you need to put bright fluorophores on weakly expressed antigens and vice versa, and avoid using or putting fluorophores that spill highly into each other on markers that will be expressed by the same cell type. Another consideration that's important is that because you're working with only 2 distinct optical paths, so the blue and the yellow lasers are collinear, and distinct from those, the violet and the red are collinear, you need to be careful in terms of what fluorophore combinations you choose. So on most modern cytometers, we're spoilt in that each of our lasers is aligned to its own optical path, meaning that, for example, you could run BV785 because it's mainly excited by the violet laser, and APC Cy7, which is mainly excited by the red laser, in the same panel. But in the image stream, you're not able to do that because the violet and the red lasers are collinear. They're not distinct in space. And these 2 fluorophores have emission profiles that are too similar to each other. It's important to titrate your antibodies and your fluorescent dyes. I mean, it's important to do that anyway to maximize your signal to noise ratio, the same as for any cytometry experiment. But because on the image stream, we don't have photomultiplier tubes where we can control the gain of each individual channel. We only have CCDs where you're not able to control the gain of each individual channel. The only control that you have over signal brightness really, once you've stained your cells, is by altering the laser power. And so you can imagine a situation where you have two fluorophores, both excited by the blue laser, and one of them gives you a very dim signal, so you turn the blue laser power up. But then when you run the second one, you find that now that signal is completely saturated because that was on an antigen that was highly expressed. And so it's important to both titrate your dyes so that you can aim for signals that are of comparable brightness, but also to think carefully and not put very differently expressed antigens on fluorophores that are excited by the same laser. We've got these two cameras, and depending on how your system is aligned, there may be a slight spatial offset between them. So if you're doing an experiment looking at the co-localization of two different probes, then it may be important for you to put those probes on fluorophores that will be collected into the same camera, because then you don't have a spatial offset between the two of them. And people often assume that the panels that work well on their conventional cytometry experiments will just translate over to the image stream. And sadly, that's not always true. They may need to be changed for the reasons that I've stated above. So what else do we need to consider? So you need true single color control. So just like in conventional cytometry, you have fluorescence spillover. And so we need to correct for that with fluorescence compensation. And when I say true single colors, the same as I would tell anybody for any cytometry experiment, I mean true single colors. So, you know, don't be bringing GFP for FITSI, and don't add DAPI to all of your single colors, as we often get. Gating controls, again, this part isn't really any different. So maybe a biological negative, maybe a mock transfected control, a fluorescence -1 control, secondary only if you're doing primary and secondary staining to help with your gating. Unstained cells are always useful to troubleshoot any issues with dyes or antibodies. And something that is maybe sometimes unique to the image stream or imaging cytometry is that you may need to consider what controls you can bring that will actually control for morphological changes. So if you can bring cells that you know have not had a shape change, for example, or their fluorescence has not polarized. So this could be including a condition at 4 degrees C to prevent cells from phagocytosing beads or something like that. And in terms of sample prep, it's important to pass your cells through a strainer to help remove clumps, although it is quite difficult to block the image stream. It has quite a wide flow cell, but it does make your life easier that you don't need to gate out quite so many clumps. It doesn't accept samples in fax tubes, so your samples must be in microfuge or Eppendorf tubes. And the sample volume is much smaller than people would typically use on most other cytometry platforms. So the minimum is 20 microliters and the maximum is 200 microliters. And because acquisition is slow, if you want a decent event rate of sort of a few hundred cells per second, then you should aim for at least 20 million cells per mil. And that scares a lot of people because that sounds like a lot of cells, but when you consider that you can resuspend that in just 20 microliters, I think that works out at about 400,000 cells in total. And you can have fewer cells than that, it's just that the acquisition may take a very long time. So again, important to titrate each of your antibodies and fluorophores when you're setting up your panel, and that concentrations that work well for your other panels may not work ideally for the image stream. Particularly, we find that viability dyes or DNA binding dyes tend to quite easily saturate the image stream. And at least to begin with, I would suggest that you limit the number of samples that you include in any one experiment. For most people, 30 samples would be a big experiment on the image stream because the acquisition takes longer. And if you have large densities of cells, then that may not be the problem. I had one user who ran 200 samples in 2 days because she was working with yeast. But I would start small. I've also run assays where 4 samples has taken all day. And finally, you should choose which magnification that you need for your experiment. And the trade-off here is only really between resolution and speed. So at the highest magnification at 60 times, the flow rate is less than half of that at 20 and 40 times. So if 40 times is good enough for what you need, most people will stick with 40 times. But of course, if you really need higher resolution, higher magnification, if you're working with smaller structures, then you need to work at 60 times. If you have plenty of cells, then you may prefer to work at 60 times anyway, because the images are bigger. And the acquisition software for the image stream is called Inspire. And this is what it looks like. And I actually find it easier training people to use this software in the image stream than training people to use, for example, Diva or Everest. And so you have a control panel on the right-hand side that gives you pretty much all the settings you need to set up your experiment. So loading and returning a sample, setting up file acquisition, turning on and off lasers. And I don't know if you can see here, but you have controls for setting the laser powers for each individual laser. Controls for magnification, so you can swap between 20, 40, and 60. Flow speed control, and some controls for focusing. And then you have a panel here for displaying your plots, so your the scatter plots and the histograms of the numeric features extracted from the images. And then at the top, you have the gallery, which shows you the images associated with the objects as they're being acquired. So each column here corresponds to one channel on the instrument. So I've got channels 1 to 12 here, and each row is one object. So these are just some beads that are running through the system, but you can see here in channel 1, this is a bright field image of these beads. And these are the same beads as these guys here. And this is the image of the side scatter. And these guys here. And this is the second bright field image. So you always have 2 bright field images, one on each camera. Okay, so data analysis. So there's a software package for analyzing your data with the image stream called Ideas. And it looks very similar to the acquisition software, except that you don't have a control panel on the right-hand side. But again, you have this plotting area here for plotting your data and gating on it, just like you would in something like Flojo, for example. And then you have the gallery at the top then for viewing the images. And you have some extra features in this software, so you can create new masks, for example, that I'll talk about in a bit, and you can create new features and new measurements about the data as well. And for most people, this software is all that you need, but you can also export the numeric data in FCS format if you want to use something like Flojo. You can export the data as CSV file, and you can also export the actual raw image files themselves. Also, there is an add-on for FCS Express for analyzing image stream data that does actually allow you to interact with the images. So a kind of typical analysis will often start by gating on the singlets. And we do that by, usually one way to do that is by comparing the area of one of the bright field images. And we use bright field because it doesn't depend on the cells being positive for your fluorophore, against the aspect ratio of the bright field image. And the aspect ratio takes the narrowest part of the image and divide it into the longest part. And so if you have nice uniformly round cells, they have a high aspect ratio, closer to 1. And where you have a smaller aspect ratio and a larger area is where your doublets tend to sit. And the nice thing about the image stream is that you as a human can use your eyes and you can decide whether a population is in fact what you think it is. So are these singlets, are these doublets, etc. And as I mentioned earlier on, if you see something interesting here, so say I was interested in a data point down here, I could click on that data point and it would immediately show me the images associated with that cell, and the other way around. So say I draw a gate around my singlets and I ask the gallery to only show me those cells, and I see a doublet in there, I can click on that doublet and see exactly here where that data point falls, and that will help me decide whether I want to change or update my gate at all. A step that is common in most analyses that people maybe won't be familiar with from conventional cytometry is that you have to gate on the cells that are in focus. So because the cells are being focused through the flow cell using hydrodynamic focusing, probabilistically, some cells will pass through the middle of the focal plane, but some will be either closer or further away from the objective and will be slightly blurry. And so we can use a feature called gradient RMS to simply gate on the cells that have a nice crisp edge versus those that have a blurry edge. And this is particularly important if you're doing anything that involves edge detection, such as gating on or identifying fluorescence in a membrane, for example. Okay, so in any one image, the image is rectangular, not circular. And often the majority of the pixels in that image are nothing to do with the object that you're interested in. And so the software will identify the pixels that it believes contain something or an object of interest. And this is called a mask. So then all of the features or the parameters that we extract from that image will only be calculated using the pixels inside the mask. And the default mask does a pretty good job for simple things like gating on intensities of fluorescence. But sometimes we may want to mask on particular regions within the cell. So it could be that we want to create a mask only for the nucleus because we want to see whether our probe is present in the nucleus, or we could create a mask for the plasma membrane, for example. The software has a nice way of you being able to create your own masks, and you can create some very complicated masks. One very simple example I'll show you here is at the top. So someone has created this mask A by taking the default mask and just dilating it out by 1 pixel. So just sticking 1 pixel on around the outside. They created mask B by taking the default mask and eroding 3 pixels. So kind of shaving off 3 pixels around the outside to make a smaller mask. And then they finally make a membrane mask by saying, take mask A and not B. And so you end up with this sort of donut where you've got A and it's cut out B. So then now we can ask for only the fluorescence intensity of a probe for this mask, and it will ignore all of the pixels in the middle of the cell. And there are lots and lots of options and ways of creating masks in the software. And you will tend to get to know the ones that work well for a particular task, but by and large, it's a case of trial and error. And creating masks is usually the step that takes the longest because you need to create one and then validate whether or not that actually generalizes well to the rest of your sample. So there are lots and lots of features or measurement parameters that are extracted from the images. And there are ones that come with the software by default, but you can also define your own. And one of the common ones is area. And the area is simply the number of pixels included in the mask expressed in microns, depending on the magnification that you're imaging at. And of course, people will sometimes quote at you that they use forward scatter as a measure of cell size, which of course it isn't. And actually cell area of the image is actually a much better measure of cell size than forward scatter. We have some features that allow us to quantify or discriminate cells based on their shape. And so aspect ratio is one that we've already seen that helped us identify singlets versus doublets. So it's the skinniest axis divided by the longest axis. And there are some others like the length, which will find the longest path through any object. And things like the minimum thickness, the maximum thickness, and this shape ratio, which, for example, is a difference between them. And there are many more morphology features that allow us to quantify changes in shape between samples. And of course, we have intensity, and this is the measure that is most like the intensity measurements that you would interpret from a conventional flow cytometry experiment. And so this is calculated by taking all of the pixels in the mask, adding up their intensities for a particular channel, and then subtracting the background. And for example, here's an example comparing the intensity of CD45 against side scatter, and you can see that you can discriminate your populations based on their fluorescence and their scatter, just like in a conventional cytometry experiment. So a kind of summary of a workflow of an experiment for imaging cytometry. So once you've acquired your samples with your appropriate settings, so your laser powers and your magnification. You perform your fluorescence compensation. And you may want to start by using total fluorescence to gate populations of interest. So what I mean by this is you may gate on whatever is DAPI negative, CD45 positive, and CD3 positive. So just gating on populations based on the total cell fluorescence, just like in regular cytometry. So at this point, we've used no spatial information at all. And then you may use some of the spatial information or the morphology measurements to discriminate different populations. So this could be gating on different cells based on their size or how elongated they are, just like you might do in an image analysis. And then depending on what you're looking for, if you're interested in studying something that only happens within a certain compartment of the cell, so maybe the nucleus, the cytoplasm, or the membrane, for example, then you will want to generate some kind of mask that faithfully captures just that portion of the cell. And this is the step that usually takes the longest. The way that most people do it is by creating a mask, applying it to their data, checking that it works well, and if not, iteratively improve on the mask. And this is the bit that takes a long time. But once you've got a mask that works well, then you can use this to generate new features that will resolve your populations. So you could use a mask, for example, on just the nucleus, and you find that that helps you discriminate between populations of cells that have high translocation to the nucleus versus low. And then often the final step is to generate some kind of score for each cell that will score that phenomenon that you're interested in. So it could be generating an internalization score or a co-localization score that you can then plot as a histogram for that sample. And then perhaps then you take the mean or the median of that score across all those cells as the value for that actual tube that you ran. And then you can compare that between your different treatment groups and run your different statistical analyses on. And so the analysis can be quite long and quite complicated, but there are some tools to help you. One of them is the feature finder, where you, by eye, perhaps you can see that there are two interesting cell types in your sample, and you manually tag the images. So you go through the images and maybe you manually tag a dozen or 2 dozen images from each of those different types of cells. And in the wizards, you use the feature finder, which will look at all of the features that the software has available to it, and it will find the ones that best separate or best discriminate between those two ground truth populations that you've given it. And hopefully then that will tell you features that will allow you to gate on those two populations. Sometimes that doesn't quite work, particularly if the cells are not all that different to each other. If it's quite a subtle difference, then there's a new option called the machine learning plugin, which sadly you do have to buy, but it does a similar thing, but in a slightly more sophisticated way. So you tell it the ground truth populations, and it will create new features that are combinations of the existing features that will help discriminate those populations of interest. So here's an example of where I did this in a file that had, I think, 7 or 8 different populations, and it found new features that will help to separate some of these populations away from the others. So there are a lot of example applications that, for example, Luminex has on its website. I'm going to take you through or give you a few examples of some that we've done in-house. So this is an example of an internalization experiment where we have some cells that are labeled with CD45, and you can see that this is kind of indicative of what membrane staining looks like on the image screen, where you have this sort of coffee stain or coffee ring stain where you have this bright ring around the outside and then maybe some diffuse staining in the middle. So our CD45 are Hirsched for DNA and some probe that they're interested in studying the internalization of. And they incubate the cells with this probe for different lengths of time and then run the cells at 15 minutes, 1 hour, 3 hours, and 18 hours. And they can see that over time, the probe goes from being on the surface of the cell to actually being internalized. And it's quite simple actually to gate and to discriminate cells that have the probe on the surface versus on the inside of them. And for every single cell, the software can generate an internalization score to score basically how much of the probe of interest is inside the cell. Here's a similar example, this time doing the similar thing, but in B cells and in T cells. And they were interested in whether there's a difference in internalization of this probe between naive B cells and activated B cells, and naive T cells and activated T cells. And so in the B cells, we have CD19, CD45, Hirsched, and the probe of interest. And you can see that as the cells are activated, we get a greater level of internalization. And the same is true for the T cells. And what's quite nice here is that actually we didn't need an additional probe to gate the activated versus the naive cells, because the size difference is so striking. It was quite easy to simply gate on the different cell types based on their cell size. Here's an example with nuclear translocation. So we've got two groups, control and knockout for some gene of interest. And we have the nucleus staying with DAPI and a probe against Notch1. And you can see that in these cells, quite a lot of the Notch1 signal sits on top of where the DAPI signal is. Whereas in the knockout cells, we find that the Notch1 signal is excluded from the nucleus, or largely excluded. And we can get a co-localization score for these two probes, and use that as a way of summarizing basically how much of the Notch1 image is in the same location as the nucleus image. And so for the control cells, we've got a higher co-localization score compared to the knockout. And for each of these samples, we could take the mean or the median of these values and use that as the summary statistic for that particular tube. Cell cycle analysis. So these are some cells that are stained importantly with PI to stain the DNA. And we can quantify the spot count, so the number of individual spots of fluorescent in the image, and also the aspect ratio of that PI image. And when cells are in prophase, their PI image is relatively round. As they move into metaphase, the image is now elongated, and so the aspect ratio gets smaller. So the cells, as they move from prophase to metaphase go from high aspect ratio to low aspect ratio. And as they separate their chromosomes and enter anaphase, we now get two independent dots or spots. And so the cells move from here to here, where they go from one spot to two spots. So it's quite nice to be able to gate cells in different stages of mitosis. Small particle analysis. So because of the time delay integration, the image stream is surprisingly sensitive for dim fluorescent signals. And so there have been quite a lot of papers showing it's a good system for analyzing small particles, although they do need to be fluorescently labeled. And there are a couple of advantages in addition to this. So one of them is that you can gate on your single vesicles. So because these things are so small, it's very easy to get multiple of them in the interrogation point at once. And using traditional PMT pulse height area width to gate out the doublets is nearly impossible on conventional cytometers, whereas on the image stream, we can gate the images and we can confirm that we're looking at singlets. And the other advantage is that it's often desirable when people run small particles to get an estimate of the concentration of them. And because the image stream is volumetric, you can simply get the number of objects per mil for your vesicles. And so finally, another example is that we can quantify changes in cell morphology. So these are, I think, lymphocytes staying with CD43, CD45, Hirsched, and then of course we have the overlay. And it's quite easy to gate cells that have a uniform shape and uniform distribution of CD43 versus those that have a capped morphology, where the cells are slightly misshapen, but importantly where the CD43 is now polarized to one edge, versus cells that have a fully formed uropod, where the CD43 now is exclusively inside the uropod and the cells are now much, much elongated. And it's really quite easy to identify and gate these different populations of cells using the image stream. And so in summary, imaging cytometry provides the spatial information of microscopy, but allows us to generate those statistically robust sample sizes of conventional flow cytometry. The panel design and sample preparation considerations are slightly different to conventional cytometry. And your data analysis is all based on numeric features that are extracted from the images. And of course, you can use masks and create new features based on those masks to suit your needs. We can use conventional cytometry, measurements like scatter and fluorescence, in addition with information about cell shape, size, and probe location. And we can define masks to measure specific areas of the cell as we need to. And any application for which the image stream's resolution is sufficient, any application that requires spatial information and the acquisition of large numbers of cells is likely to be a good candidate for the image stream. So that was a brief introduction to imaging flow cytometry. I hope that was useful, but I am happy to answer any questions that you've got.
Excellent. Thank you, Kevin. If you just go to the next slide, so we can leave that one up while we're asking questions, answering the questions here. Just to say that if you want to see what is coming up from the CRIC, if you go to the What's On page, everything is listed there. You may also want to follow both the Royal Microscopical Society and Flow Cytometry UK, both of which have a lot of educational offerings at the moment. And as having said, if you've got any questions that we don't ask now or answer now, please feel free to contact us anytime you like. And at the bottom there is the link to the YouTube playlist, which has a lot of these webinars already up there. So there are a few questions that have come in the Q&A. I think the first one you've covered, Kevin, but it may be worth just reiterating it, in the fact that in normal flow cytometry, we start with forward and side scatter, which of course we don't really get on an image stream. So, how do we define where our cells are? What's our first level of gating?
Yeah, so a lot of people, when I'm teaching them the image stream, struggle with this actually, because they're so used to starting with forward and side scatter. So I think most people end up starting with their singlet gate usually, which will be the area of the bright field image against the aspect ratio. But often what I will do, and I think people find this useful, is I'll sort of emulate the forward and side scatter plot. So what I'll plot is the area of the bright field, which is cell size, against the side scatter. And that helps identify the cells from either debris or the beads that are run as a consumable through the system. So there's no forward scatter at all. So either you can use cell area, or you simply jump straight in with your singlet gate.
Okay. This is a sort of practical question about if you've got a long acquisition, how do you make sure your cells are kept happy?
Yeah, so you can run fixed cells. So if you do a time course or your treatment, and then you fix all the cells together at the end of that, and then you can run them sort of freely as you need. We have some people, once they've done one experiment, they kind of know how long sample acquisition will take for that experiment. They might stagger their treatments so that they will come off one by one in time. Otherwise, if the timing isn't super important, often we will keep the samples in the fridge, but ideally you keep everything the same for the experiment. And the sample acquisition doesn't always take that long. It's just if you have a limiting number of cells, sometimes sample acquisition takes only a few minutes per sample. It's just the ones that it takes half an hour, that becomes an issue.
And in terms of compensation, is the sort of data spread that we see in fluorescence, normal conventional cytometry, similar in imaging cytometry? Do we deal with that in the same way?
So the data spread actually is, I often find, is actually a little bit worse than you would get on a conventional cytometer. And it similar considerations in that the more two of your fluorophores overlap with each other, the worse the data spread will be. And so it's just a case of trying to pick fluorophores that are far apart from each other, or putting them on antigens that are mutually exclusive between cell types. One interesting thing with the image stream, of course, is that when you get compensated data and you end up having negative intensities, of course, what that actually means is that you have pixels in the image that are below background. So if you have an awful lot of data spread, what you can actually have is images where you've got a sort of a faintly colored background in the image and a black burnt out hole where your cell should be. But other than that, the considerations are the same. One other thing with the image stream is that the cross beam spillover tends to be higher than on most modern conventionals. So what I mean by that is, for example, it's quite normal to see a bright DAPI stain spreading over into your FTSE or your GFP channel.
I suppose it would look different anyway because you're not using the same filter sets, are you, on the image stream than you are on a cytometer.
Yes.
In most cases.
Yeah, absolutely. Yeah.
Okay, so there's a question about analysis here, about the variation in size of cells and in the size of the cells or the shape. Would that mean you'd have to mask individually on each cell?
I think you'd go mad if you did that. So yeah, so the way that I didn't explain that so much, but the way that you set up the masks is you sort of create a rule that is kind of applied then to every single cell. So the rule could be mask all the pixels that are between two different intensity values, or it could be a rule such as take the default mask for that cell, shrink it by a certain amount, and then combine that with a knot of another gate, for example. So you kind of define a rule, and the software will apply that to all of the cells. And then what I usually do is look at the other cells and make sure that my mask has generalized well to it.
Okay.
Mm-hmm.
And you talked about the size of cells. Is there a limit then to the size of the cell? So if you've got something like a neuron or a sperm cell, for example?
Yeah, so the image stream is a little bit weird like that, because actually technically there isn't really an upper limit for length. I mean, there probably is in terms of software, but if you reach that, what you're looking at is not a cell. So because things orient lengthways in the axis of flow, you can image some very long objects. In terms of the width of the image, at the highest magnification, I think the widest object you can image is 40 micron wide. And at the lowest magnification, I think you're on to something like 150 micron wide. So it's got a big flow cell. So I've not ever seen anybody struggle to image large cells. It's just whether if you're trying to image something larger than a cell, I don't know, maybe an embryo, you might need to consider that. But if you're looking at neurons, that's fine.
I suppose one question that we always get asked with the image stream is, why doesn't it sort?
Yeah, I think the Luminex people are bored of us asking that, actually. Yeah, I think it would be a mean feat for them to create an instrument that had the processing power to do that.
Exactly. I think you mentioned those simple sorters, and they are very simple, aren't they? They're microscopes that pick out one color.
Yes,
which is much simpler than the way this works.
Yes. Yeah. Sadly, we don't have an imaging sorter that is high performance like the image stream.
Excellent, Evan. So I think we've come to the end of the questions there. As I said, if anybody thinks of anything a little bit later on, please feel free to email us. But if there aren't any more questions, thank you all for coming, and hopefully we'll see you again soon. Thanks very much. It's goodbye from us.
Bye.